Yeah, myself Dr. Vaibhav Chaudhary. I am a practicing medical oncologist in Wockhardt Hospital, Nagpur, and I expertise in mostly solid tumors. Lung is something which is very close to my heart. Actually, everyone's lung is very close to their hearts. So the newer targets in lung cancer space, actually something which has been very, very exciting in whole oncology space is lung cancer. And it has come a long way in past 20-25 years. Courtesy chemotherapy, targeted therapy, immunotherapy, and the ADCs, antibody drug conjugates. These are the new kids on the block, and on, newer things are coming on. So my job today is actually a very simple one. I just have to discuss about three targets, which obviously I was not able to pronounce properly. One is CD73, one is NKG2A, and one is TROP2. These three markers actually are very, very important for filling the gaps in today's continuum of treatment of lung cancers in various stages as such. Like we are very, very happy, and very happily we tell people that we have got target therapies and we have got TKIs, and it can take care of your cancer for years together. Yeah, true. But there comes a time then even after the TKIs, patients have progressed, and then honestly speaking, there is patient is still having a good performance status and he doesn't have any standard line of therapy left. So it is left at the mercy of treating medical oncologist. Whatever his experience based treatment is there, he has to give. Or then the best thing which is possible is enrollment in a clinical trials of these new evolving drugs. So gradually we will start deciphering these three targets, and these three are actually very, very important. In the recent ESMO 2021, very good data were presented targeting.
Sir, sorry for intervening. Your slides are not moving. Sir.
Yeah, now it is.
Yes, perfect.
Yeah. So antibody drug conjugates. First of all, I'm starting with TROP2. Now TROP2 is a tropoblastic antigen 2. It is also called as epithelial glycoprotein 1. It is actually a very good target which can be actually exploited for giving targeted therapy, because it is a transmembrane protein with extracellular domain, and it is significantly overexpressed in a variety of cancers, not only lung cancers, but a lot of tumors, especially lung cancers, all three varieties, squamous, adeno, as well as in small cells. So it has been studied extensively. Overexpression of TROP2 is also very common in metastatic breast cancer as well. It has been said that its overexpression is associated with poor survival rates in patients. So all of us remember this molecule, sacituzumab zoviotecan. This actually works on the same target, TROP2, but the earlier trials did include a few patients of lung as well, but they found a very good signal in triple negative breast cancer. And as it is an ADC, so in triple negative breast cancer, post 2 lines of chemotherapy, it has just recently achieved 1.5 years back the FDA approval for treatment. On similar lines, the drug which is much more active in lung cancer, that is datopotamab deruxtecan, DXD we can call it. So this is the molecular structure and this is the mechanism of action of this ADC. It is just like the first ADC which we came across was in breast cancer, that was TDM1. And it is very simplistically written here that, yeah, it does bind to that transmembrane TROP2 and get internalized into the cancer cells. Obviously, they are overexpressed on cancer cells. It goes inside the cancer cells, and then it contains an active chemotherapy moiety, that is DNA topoisomerase 1 inhibitor, and it causes DNA damage and apoptosis. It doesn't only stop there. Actually, it permeates and penetrates to the neighboring cells, and it releases the payload there as well. This is because of that cleavable linker. So these are actually more advanced molecules, ADCs, than the first generation ADCs which came in. And that is the reason that these are having a very profound effect. It takes care of tumor heterogeneity. It takes care of resistant cells as well. So that's why it leads to a marked tumor regression. This is about the mechanism of action. So not actually, it is this molecule is being studied upon, and we just received recent updates from this phase 1 global study of Tropion Pan Tumor 01. Actually, it is a study which is devised out of necessity, and it is an ongoing multi-center open label dose expansion study of Dato-DXd in solid tumors, almost 210 patients, which actually included some lung patients as well, lung cancer patients. So the objectives were assessing safety, pharmacokinetics, anti-tumor activities, and the patient who have actually progressed after standard treatment and had no standard treatment available with measurable disease. Stable and treated brain mets were also permitted. Patients were not selected based on TROP2 expression or age. So these are the results in short, that 34 patients had advanced metastatic NSCLCs with actionable genetic mutations were taken in these studies. Out of these 34 patients, 8 patients were maintained at a dose level of 4 mg per kg, 10 patients at 6 mg per kg, and 16 patients at 8 mg per kg. The protocol of phase 1 study, decent number of patients were there. 82% of the patient had received more than 3 lines of prior regimens. As we know, lung cancer as well, and also these biomarker positive lung cancers do receive TKIs. 85% had prior TKI exposures, and almost 69% of EGFR had prior received osimertinib. So these are those patients who had progressed after years of TKIs. Median duration here was 13 months, and this was actually very, very amazing and surprising to many people that the confirmed ORR was almost 35%. It's a big number in such a refractory setting, and median duration of response was 9.5 months. Most common any grade adverse event was nausea, 62%, stomatitis, hematological toxicities were infrequent. One alarming signal was there that one patient actually had a grade 5, I mean, patient died with interstitial lung disease, and he was at the top dose level of 8 mg per kg. So now people are titrating the dose to 6 mg per kg, and then based on these results, we can draw some conclusions. It's actually very early, phase 1 studies are like that, but anti-tumor activity and safety in advanced metastatic NSCLC patients with AGAs are encouraging. Actually, they don't have a current standard line of treatment. Ongoing phase 2 Tropion 05 lung trial actually is assessing the lower dose levels at 6 mg per kg in such patients, and it is an ongoing study. Time will answer whether this molecule is successful or not. So nonetheless, interesting data. Coming to our next topic, that is anti-CD73 and anti-Nkg2A immunotherapy combinations. This is a COAST trial. These are again presented in ESMO this year, and this is an open label phase 2 platform. Durvalumab alone in combination with novel agents in patients with locally advanced unresectable stage 3 NSCLC. As we all know that Pacific 3 has actually established the new standard of care in stage 3 lung cancers. But what happens after that? Not everyone is cured with Durvalumab. As we all know that 42.9% remain alive and 33.1% rely at the and PFS of 5 years. Additional immunomodulation is required because obviously, though Durvalumab is very, very active and has added a lot of years to life of such patients, but still at the end of 5 years, almost half the population is gone. So to add on to that, this trial actually was initiated, and newer drugs were tried in combination with the standard of care. So this is the rationale. Rationale is very, very interesting that after CTRT, which is the standard of care for unresectable stage 3 NSCLC, radiotherapy induces expression of CD73 and Nkg2A ligand, which inhibit anti-tumor immune responses. So oleclumab, which inhibits CD73 to reduce extracellular adenosine production and thereby promoting anti-tumor immunity. The patient actually it has shown some good results in phase 1 study of advanced EGFR mutated lung cancers. So this is a rationale. Second is monalizumab blocks Nkg2A to reduce inhibition of NK and CD8 positive cells. and it did show a positive signal in a study of head and neck cancers. So combination of radiotherapy and these two molecules is also taking place, and that also is having good results in preclinical models. So this is the COAST-2 study, a very simple design where they had actually randomized in 1:1:1. One is the standard of care durvalumab arm. In second arm, they have added oleclumab, and in third arm, they have added monalizumab with a primary endpoint of ORR by investigation assessment. Secondary endpoints of safety, DOR, DCR, PFS, OS, PK, and immunogenicity. So they wanted to actually have 60 patients in per arm. As between July 19 to July 20, they enrolled some 186 patients, and they achieved their target of enrollment. In May 17, 2021, all patients had a minimum of 10 months potential follow-up. So this data was presented where, as you can see, the baseline characteristics are almost similar. They had almost equal percentage of squamous as well as non-squamous histology. They had more patients of they had actually a lot of PDL1 status was unknown, as you can see, but that was there in all the groups. So the groups were equally matched. Cisplatin versus carboplatin was also there, which actually in subset analysis it will come up. And the patient which with anti-tumor activity by investigator assessment was there in ITT population, and confirmed ORR was actually better in the combination groups, in both the combination groups of D plus O as well as D plus M, were better as compared to durvalumab numerically. Similar things were observed in form of DCR at 16 weeks and the median DOR as well. These are the PFS curves, and here, as you can see, that there is a marked PFS improvement with both the combinations actually, and as compared to the baseline of durvalumab. At the 10 months, 39% of the patient of durvalumab were disease-free compared to 64 and 72%. So this is a subset analysis. As you can see, across the board, almost all the categories of the patients, the combinations were better individually as compared to durvalumab. When it comes to the safety, as you can see, the class effect of durvalumab and the safety profile, we are all very well versed with it. That was seen in every in the combination group as well, and there were no alarming toxicities. There were no new toxicities which are attributable to these two drugs when given with durvalumab. We are very well versed with managing the immunotherapy IO toxicities, and as you can see, it was all very predictable and manageable. So as you can see, there was no alarming toxicity, as I told in the last molecule, that ADC, which had interstitial lung disease and patient died. Here in this study, there was no alarming toxicities like that. So what it leads to is actually it is very encouraging study, which shows that by adding these two drugs with durvalumab, either of them actually had a better response, and actually both the combination numerically increased ORR and significantly improved PFS over durvalumab. PFS benefit was with both combination was observed across subgroups, across the PDL1 status as well. So this is one more concern, that even progress after this is not pertaining to this setting, but in stage 4 setting, patients do progress on PDL1 inhibitors, and then these drugs may actually give a ray of hope to such patients. So these data actually support further evaluation of these combinations in registration intent studies. That's all. I think I don't have more data on these newer drugs, so I would like to take a leave, and thank you. Thanks a lot.